Okay, so now we are going to be talking about aerobic and anaerobic metabolism of glucose, which you might say, well, we've kind of been talking about that anyway when we're talking about glycolysis and then running through the Krebs cycle, and you would be completely right. But now we are going to be looking at those not from the metabolic pathway point of view that we have been really looking at the different chemical reactions and transport mechanisms of the molecules, but we're going to be looking at the energy portion of this and kind of getting into the detail, not kind of getting in, totally getting into the detail of how much energy we're making during glycolysis and during the Krebs cycle / electron transport chain. And remember, now we are just focusing on glucose, so carbohydrates, and we are looking at the energy from that. But also just remember that we can use lipids and proteins as sources of a substrate for the Krebs cycle for energy creation. But here we're just going to maintain our focus on glucose for now. So glucose is a source of quick energy for cells. So when we are doing glycolysis, breaking down that glucose, it happens very quickly. So we can get energy out of our glucose really fast. So it's a good thing to use if you have a need for quick energy, like if you're exercising in a kind of vigorous sort of way and your cells need a lot of energy fast. Using glucose as the substrate for energy production is a good thing to do. Now, just to review, aerobic means oxygen present, anaerobic means oxygen is not present. So typically, hopefully, you have oxygen present, but if you are exercising pretty intensely, or there's other situations, you're in a low oxygen environment, or carbon monoxide poisoning would cause you to be in a more hypoxic, which means low oxygen, or anoxic situation, that could, mean cause you to not have oxygen available. But typically when we're thinking about normal situations, the only time you'd be in anaerobic respiration would be if you were just exercising so much, or you're making so much energy, using so much oxygen that you used up all your oxygen faster than what you can breathe it in. So rather than something kind of extreme like these two. But typically we'll be in aerobic respiration, which is more efficient. You make more energy, and there aren't kind of toxic byproducts that come along with anaerobic respiration. Now, we have seen the chemical reactions that create the energy, and we've seen the electron transport chain. So we've seen glycolysis, we've seen the Krebs cycle, we've seen the electron transport chain. Here are just pictures of glucose. So this would be a stick diagram of glucose, this would be the ring diagram of glucose, and then an actual model of the glucose molecule. But we can list out the steps in the chemical reaction of glycolysis and the Krebs cycle as this. So C6H12O6, that's glucose. Then in the Krebs cycle, or the electron transport chain I should say, we're going to use 6 oxygen, and that's going to give us a yield of 6 carbon dioxides, 6 water molecules, and then energy as ATP. I didn't put the number here for a reason, because we're going to get into the exact calculation of how much energy we are going to get out of our glucose. Now remember also that when we do glycolysis, so I'm not going to write out the whole, all the steps of glycolysis, but we are getting 2 ATP and 2 pyruvates, or pyruvic acid. So everything that happens in the Krebs cycle is going to be happening twice because of the 2 pyruvates. We are also getting NADH out of glycolysis, and then again out of the Krebs cycle, and FADH2 out of the Krebs cycle to go to the electron transport chain to be used to create the energy. And also remember that those pyruvates, or pyruvic acids, are going to be converted into acetyl-CoA to enter the Krebs cycle. Okay. So in order for you to go from glycolysis, which is anaerobic, to the Krebs cycle, which is aerobic, you have to be able to turn your pyruvate into acetyl-CoA. Now, this takes an enzyme called pyruvate dehydrogenase. There's that dehydrogenase again, but this time we're talking about pyruvate. And pyruvate dehydrogenase is going to turn your pyruvate into acetyl-CoA. But it requires something called a cofactor. And a cofactor is just a molecular substance required for an enzyme to work. So without your cofactor, the enzyme won't work either at all, or maybe it just won't work as well. And a main cofactor for pyruvate dehydrogenase is thiamine, which is vitamin B1. And it's a big cofactor for pyruvate dehydrogenase. So without vitamin B1, pyruvate dehydrogenase can't effectively turn pyruvate into acetyl-CoA to go from glycolysis, the anaerobic respiration part, into the Krebs cycle, the aerobic respiration part. Now, cofactors often are vitamins. We'll see various cofactors throughout the semester, especially as we get into like antioxidants and things like that, which are also vitamins. So often also vitamins, I should say, not always, but we'll see various cofactors as we move through the semester. But thiamine is an important one for pyruvate dehydrogenase. Also other B vitamins like niacin, there's thiamine, riboflavin, other sort of fatty acid things, lipoic acid, pantothenic acid. These are all other cofactors that are important during glycolysis through the Krebs cycle. So it is important to have these cofactors present in order to allow those things to happen, or else you get stuck just in glycolysis and anaerobic respiration, not getting your total amount of energy out of your glucose. Okay, so how are the cells getting the glucose? So one, you eat carbohydrates. They are broken down into simple sugars to be absorbed in the intestinal epithelium. Here I just said they're broken into glucose, but we could break them down into other simple sugars as well, so that we can absorb them into the intestinal epithelium and then into the blood. But we're just going to keep this simple and just look at glucose being absorbed. Other simple sugars could then be converted into glucose or used as a substrate starting into glycolysis, but we're just going to keep it with glucose for now. So our glucose has gone into the blood, and then it can circulate and enter into other cells to be used directly as energy. Now, some cells, specifically hepatocytes, remember hepatocytes are liver cells. They do a lot of stuff. But one of the things that they can do is gluconeogenesis. That is making new glucose. Okay, so when you are making new glucose, you are taking something, some other molecule, and turning it into glucose. It is a separate metabolic pathway that breaks down whatever molecule we're talking about, and it converts it into glucose so that you can have glucose. And this is important for when you are not, eating or digesting, like what was called the fasting state. rather than the fed state, so as opposed to the fed state. So in the fasting state, you haven't eaten for a little while. This isn't referring to you're on a fast, although, if you are on a fast, you are in a long-term fasting state. But every day, you're going to be going into a fasting state. If you eat dinner at whatever time you're eating dinner, and then, you don't eat until you go to bed, maybe, and then you go to sleep, and you sleep for however many hours you sleep, that glucose from dinner is not going to last you all night long until you have breakfast. So you're in a fasting state overnight. Anytime you haven't eaten for, it depends on the meal, so 4 to 6 hours, say, you can start to enter the fasting state. And this is where you don't have glucose coming from your intestines going into your blood. So you need to do gluconeogenesis. The fed state is when you've actually already, you've eaten and you are currently absorbing glucose. So you are getting glucose into the bloodstream from the food that you ate. And I actually encountered this. I was on vacation and last week, and I just found this, kind of health food store, and they had all these cool different fruits and stuff available, and one was a lychee, and I wasn't quite sure how to eat the lychee, and so I was looking it up, and I just stumbled accidentally across this article about how lychees, if they're not completely ripe, they have an enzyme that prevents gluconeogenesis. So you don't want to eat an unripe lychee if you're in the fasting state, because then you can't do gluconeogenesis. But if you are in the fed state, then you're fine to eat the lychee even if it's not ripe. And if it's ripe, it's totally fine. I just thought that was kind of interesting and funny that it sort of links up with that, but it's just something that would block gluconeogenesis. Now, why you need to do gluconeogenesis, why it's so critical that you do, is that you can do gluconeogenesis in your hepatocytes. You can do that in your skeletal muscle a little bit in the kidney cells. Maybe astrocytes in the brain and spinal cord, maybe they can do it. That's not 100% certain yet, but there's some evidence that suggests that possibly they can. But cells like your brain cells can't do gluconeogenesis. Now, other kinds of cells also can't do gluconeogenesis, but what they can do is use something else for energy, such as a lipid or a protein. But the brain cells, so your neurons, I should say, or not just brain cells, neurons, require either glucose or keto acids for metabolism. So if you don't have glucose available or keto acids, your brain cells are in trouble. So gluconeogenesis being done predominantly by the hepatocytes is important, especially for your brain cells, which are really reliant on that kind of thing. Thanks to the hepatocytes, they need those hepatocytes to be making glucose for them to be able to function when you're in the fasting state. Now, when you're in the fed state, so you've eaten and you have all kinds of nutrients coming in from your intestines into your blood, so you have extra glucose molecules. You can bind glucose molecules together and form glycogen, and that's called glycogenesis. So we saw gluconeogenesis, making new glucose. Here we have glycogenesis, that's making glycogen. And here is glycogen. C24, so 24 hydrogen, or 24 carbons, 42 hydrogens, and 21 oxygens. So we just took a bunch of glucose molecules and strung them together That's glycogenesis. It's its own metabolic pathway, and we will get into that metabolic pathway soon. But it's essentially the stored form of glucose. And then we could break that down. So glycogen can be broken back down into glucose when needed. And that would be glycogenolysis. So we have gluconeogenesis, making new glucose. We have glycogenesis, making glycogen. And then we have glycogenolysis, breaking down the glycogen back into glucose. So this is where our glucose is coming from. Okay, now once we have our glucose, we have two opposing hormones, insulin and glucagon. Insulin takes the glucose out of the blood, puts it into the cells. Insulin is made by your beta cells in your pancreas. It's released after you've eaten and when you are in the process of absorbing glucose, so that you can take that glucose and put it into your cells. Glucagon is the reverse hormone, and it is part of the thing that causes glucose to leave cells, in particular your fat cells where we can store extra glucose. There are other factors that cause glucose to leave the cells that we're going to get into later in, I believe it's Unit 3. So we'll talk about other things. So just kind of put a pin in that, think, okay, we're going to amend that a little bit later on in the semester. But glucagon is something that is also made in the pancreas, not pancreas, so it's made in the pancreas, but it's made by the alpha cells in the pancreas, and that would help you to break down fat, so taking the stored glucose out of the fat, putting it into the blood, so that then the glucose can be used by other cells. Okay, so we need to look at how much energy each molecule of glucose. The old way of teaching this was to just say 38, and that is what everybody used to think. But that has been refined a little bit, fine-tuned, made more accurate because of something called a PO ratio that has been corrected for NADH and for FADH2. Now, what is the PO ratio? That's over here. It's really just talking about how much energy you're getting out of each molecule, whether we're talking about starting the electron transport chain with NADH or if we're starting the electron transport with FADH2, how much actual energy are we going to get from those molecules from running them through the electron transport chain. Technically, it means the phosphate to oxygen ratio. Boy, I'm full of typos. Ratio, not ratio, that's not even a word. So the phosphate to oxygen ratio. So with that PO ratio, you're going to see how many ATP molecules you get every time you oxidize NADH or FADH2 and bounce their electrons down the electron transport chain until we hit, the final electron acceptor, which is oxygen. So that is going to create the ATP in the end through that ATP synthase molecule, if you remember that from the electron transport chain. That's the end of the electron transport chain, or the end result of the electron transport chain. You could say the oxygen is the end of the electron transport chain, but it drives hydrogen across the membrane, and we use the ATP synthase to create ATP from ADP. Okay, so that PO ratio is just saying how much are we going to get from either NADH or FADH2. So for NADH, the PO ratio is 2.5. For FADH2, it's 1.5. So they used to think NADH was 3 and FADH was 2. It's close, but not exactly correct. So it just changes everything just a little bit. What we end up with is 31 ATP for every glucose molecule. And we're going to go through the math and see where that number comes from. It's not just the PO ratio, that's a big part of it, but there's also, you know, what's coming out of glycolysis, what's coming out of the Krebs cycle itself rather than just the electron transport chain. And also, are we using energy at any point? Are we using hydrogens at any point? Because remember, the hydrogens are what are going to be, you know, used to use the electrons or to move the electrons down the electron transport chain. So if we're using a hydrogen, kind of taking it away from the electron transport chain, that reduces how much energy we're getting at the end. So we're going to do the math on this and see where all this energy is coming from and where some of it is going. So how many hydrogen does it take to make one ATP? It takes 4 hydrogen to make one ATP, so 4 to 1. So not a lot of hydrogen, 4. The hydrogen ions are coming off of NADH and FADH2 as we're going through the electron transport chain, oxidizing and reducing, oxidizing and reducing. We are using those hydrogens to move across the inner cell membrane and to drive that ATP synthase to create the energy. Okay, so here is a picture of ATP. We have adenine, that's the A, part of ATP. Here's ribose, that's just the sugar part of it. And then 3-phosphate triphosphate, so that's the TP part of it. If we were to cross one off, that would be a DP, diphosphate, adenosine diphosphate. So that would be the lower energy form. Here we have the stored energy form, ATP. So remember, when we start glycolysis, we use 2 ATP. Remember, if you think back to our first time that we saw the glycolysis metabolic map, we saw that we have to use 2 ATP at the start of glycolysis. Then we make 4 ATP during glycolysis. So we make 4, but we use 2 to start. So we only get a net of 2 ATP. Just think of it like money. You know, like if you place a $2 bet on, I don't know, the lottery or something, and you win $4, well, you have $4, but it took you $2. you already paid for $2, so you only really win $2, if that makes sense. So that's what's happening with the ATP. You use 2, you make 4, so you end up with, you gained 2 ATP, which is not a lot of ATP. So, you know, we want to get this into the electron transport chain so we can create more, so we want to, you know, do other things. But we're also getting 2 NADH molecules out of that, which we can use the malate aspartate shuttle to get those NADH molecules, or to get the hydrogen, I should say, off of those NADH molecules into the mitochondria for the electron transport chain. Okay, now assuming we have oxygen present, we can take the pyruvic acids, or the pyruvates that we made during glycolysis, and we can carry them into, or we can convert them into acetyl-CoA, which can enter into the Krebs cycle. We will make 4 NADH molecules, 1 FADH2 molecule, and 2 more ATP for every time that Krebs cycle turns. Now remember, we have 2 pyruvates, which means we have 2 acetyl-CoA molecules to enter the Krebs cycle. Oops. So we have 2 times around the Krebs cycle for each glucose molecule that we started with. So we have to double all of this, so it would be 8 NADH molecules 2 FADH2 molecules for ATP because it's happening twice. So here's our totals as of now. In glycolysis, we net 2 ATP and we get 2 NADH. In the Krebs cycle, we get 4 ATP because we doubled it, remember, 2 per round, 8 NADH, 4 per round, and then 2 FADH2, 1 per round. So before we hit the electron transport chain, if we just add up glycolysis in the Krebs cycle, we got 6 ATP. Still, not a whole lot going on there. We got 10 NADH and we have 2 FADH2. These are going to be where we're going to get a lot of energy from because these are what are going to go to the electron transport chain. Okay, now if you think back to the PO ratio, remember we said the PO ratio for NADH is 2.5. The PO ratio for FADH2 is 1.5. So you think, okay, we can just multiply that. You know, we could go 10 NADH times its PO ratio, which is 2.5. We got 2 FADH, that PO ratio is 1.5. You go, okay, well we could do that and then, you know, add on that 6 ATP. So we can do that here. So 6 ATP, 10 NADH times 25, that would give us 25 ATP. This, just hold on to that thought for a second. If we took our 2 FADH2 and times by 1.5, that would give us, my math is crazy. No, it's not actually. So, okay, we give us 3 ATP. So, sorry, my math skills are not great. So, we're ending up here, so 3, 25, and 6. If you add all those together, you get 34. We said already that we're going to get 31. So, something is wrong here, obviously. What is wrong is not obvious. But where it is in the NADH and the FADH2, and how we're not getting a perfect transfer of hydrogens over to the electron transport chain because of a couple of processes involved, which are going to use up some stuff. So we're going to kind of knock down our yield to 31, not 34. Okay, so why is that happening? Why are we losing those 3 ATP? The NADH that came from glycolysis, that's why, that's part of it. So we are having that NADH being made from glycolysis in the cytoplasm. So it's not ready to go into the electron transport chain. It has to be transported into the mitochondria. And we are going to use the malate aspartate shuttle. Now, we use the malate aspartate shuttle because we are mammals. If we were insects, we would use the glycerol phosphate shuttle. But we are not. We are using the malate aspartate shuttle. So while we're using that shuttle isn't a perfect transfer. So some of those hydrogens are getting lost. We are also losing some hydrogens as we go through the Krebs cycle, specifically when we go through the succinyl-CoA synthesase reaction, which is, here's our Krebs cycle, here's our succinic acid. That's never gonna come into focus, so succinyl-CoA synthesase reaction, oops, my arrow's not, it would be creating the succinic acid. Some hydrogens are going to get lost there too. Okay, so we are losing a few hydrogens. Now, in the loss of those hydrogens, remember it takes us 4 hydrogens to make 1 ATP. So we're going to end up losing 3 ATP. So you can actually calculate how many hydrogens you lost through the malate phosphate shuttle and through that succinyl-CoA synthasase reaction. If you're losing 3 ATP, so we have to get 3 ATP, or not get, I should say, 3 ATP, and it takes 4 hydrogens to make each of those 3 ATP. So 4 for each one times 3, we are losing, oops, why is that showing? There, 4 times 3 equals 12 hydrogens lost. So that's the ATP lost, or not gained. We lost 12 hydrogens in the malate aspartate shuttle. And in the succinyl-CoA synthasase reaction, okay, so those 3 ATP lost, our total that we got from back here, which was 34, we got to subtract those 3, and we get our 31 total of ATP. So 31 total ATP from glycolysis, plus the Krebs cycle, plus the electron transport chain. And that's for each molecule of glucose. Okay, now anaerobic metabolism of glucose, this is where we don't have oxygen present, so we're exercising, like this person looks like they're running or something, and they have run out of oxygen, so they are stuck in glycolysis. They can't run the Krebs cycle and do the electron transport chain. So remember, in glycolysis, we are yielding pyruvate that would, if we had oxygen, turn into acetyl-CoA, but without oxygen, it ferments into lactic acid. That's when you start to get kind of muscle cramps and stuff like that. That's what's happening here. A muscle cramp, your lactic acid, your pyruvate is just fermenting into lactic acid. It's building up, and it's an acid. It's causing acidosis in the tissue, which is not great. A little bit is fine. The muscles can handle a little bit, but it's not great long term. And it can cause your muscles to feel crampy and painful. You're also making NADH, which then it can't go into the electron transport chain because there's no oxygen to be the final electron acceptor. So you've got to deal with all those things. This extra pyruvate and this extra NADH. You're getting your too little ATP, which is a little bit of ATP, but now you have this buildup of stuff. So here, an enzyme, lactate dehydrogenase, this is what is causing the pyruvate to ferment into lactic acid, and it's also taking your NADH and turning it back into NAD+. So you have now just lactic acid instead of pyruvic acid turning into acetyl-CoA. This is called, when you have too much of it, you call it hyperlactemia. Now, other causes of hyperlactemia would be anything that stops you from turning pyruvate into acetyl-CoA. So remember, we were talking about the pyruvate dehydrogenase reaction. Let me find that. Where was that? A bunch of slides ago, apparently. Here we go. So the pyruvate dehydrogenase reaction is what turns pyruvate into acetyl-CoA, and a cofactor for pyruvate dehydrogenase is thiamine, vitamin B1. So if you don't have vitamin B1, well, then your pyruvate isn't turning into acetyl-CoA, and you're going to end up with hyperlactemia again. Now, once you stop exercising or get some thiamine into your into your life, then that lactic acid can be turned back into pyruvate. This can only happen in the liver. It's through a process called the Cori cycle. It is aerobic. You need oxygen for that. So after you've finished exercising and you're, replenishing your oxygen supplies, you can run this Cori cycle, but the amount of oxygen that it takes to turn however much lactic acid you made back into pyruvate, that's called the oxygen debt. So how much oxygen you need to use to run the Cori cycle to turn that lactic acid back into pyruvate, which can then be turned into acetyl-CoA and run through the electron transport chain. Or the Krebs cycle and then the electron transport chain. Okay, so the ATP yield for anaerobic metabolism is obviously a whole lot less than it is for aerobic metabolism. So we make 4 ATP during glycolysis, but it took us 2 to get started in the first place, so our net ATP is 2. So that's just a lot less ATP than what we had from the electron transport chain. Now, if we were doing glycolysis from glycogen, so we are not just starting with glucose, we're breaking down glycogen instead. So remember, glycogen is multiple glucose molecules, and we would have to break it down. So this would be something that would be done predominantly in the liver cells. Also a bit in skeletal muscle, but not really other cells can do this. But let's say we're in a hepatocyte in the liver and we're breaking down glycogen directly into energy. We would get 4 ATP out of that process. It takes us 1 ATP to get the process started, so we get 3 ATP out of that. Again, that's not a lot. So it's not the most efficient way to do. It's good to have oxygen available to be able to do the electron transport chain. Okay, now just to introduce the next set of information, the next set of videos, we are looking here at glycolysis and doing the Krebs cycle and the electron transport chain to make energy, but we can also have other things that we do rather than just straight up glycolysis forming pyruvate, forming acetyl-CoA going into the Krebs cycle. We can have side reactions, other pathways we can take that branch off of various points of glycolysis. And these pathways are called shunts. Anytime you have a side pathway, it's called a shunt. And most metabolic pathways do have shunts, and it's all kind of determined by what enzymes are available, what substrates are available, varying amounts of the different substrates. We're going to get into all that in Unit 3. It's called metabolic mapping. But just to kind of introduce metabolic mapping as something to start thinking about, it's how a cell determines if it's using the main pathway or a side pathway instead. And that would be called a shunt. So enzymes present, substrates, so the things that you start the reaction with, cofactors that are present, metabolites too that are present. And by metabolites I mean things that are created during the metabolic pathway. So like NADH, acetyl-CoA, even ATP. These are things that are going to maybe give a feedback into like, okay, we've done enough glycolysis here, we have a lot of energy, so let's take a shunt instead and do glycogenesis instead, or one of these other shunts. Or maybe a lack of some of these things might cause you to take another pathway out of necessity because you don't have the, the ability to do. Like if you're missing pyruvate dehydrogenase and can't turn pyruvic acid or pyruvate into acetyl-CoA. So these are all reasons for having a shunt, or causing you to go into the shunt rather than the main pathway. So we'll look into how the cell decides how to do that in Unit 3. But there are some shunts off of glycolysis. We've got here the main pathway, and then you can see here at glucose 6-phosphate, you can see a pathway branching off into this thing called the pentose phosphate pathway, which we'll talk about in a little bit. The next pathway, the next shunt we're going to talk about in our next video is the 2,3-biphosphoglycerate pathway. There's a shunt for lipogenesis, so creating lipids, fats. A shunt for glycogenesis, so we know that that is creating glycogen. So we're going to look at the possibilities of these shunts in our next slide, or our next video.